Hi. So now we are moved to the last session of this course, and in this session we will continue the parasite infection. Here in this session we will discuss about African sleeping sickness, okay, or trypanosomiasis. So let's begin. So African sleeping sickness is basically caused by, here you can see that it is caused by Trypanosoma brucei, okay, and here you can see that this blue or violet color parasite, it is a flagellated parasite, okay. It's a flagellated protozoan, cause chronic debilitating disease. Debilitating means that the disease which cause you extremely weak, okay. That is the meaning of debilitating disease, okay. Characterized by unique mitochondrial DNA, lives exclusively as an extracellular parasite, okay. You have seen that this malaria parasite are present in liver cell, they are present in red blood cell, but here it is unlike malaria, they are present only in extracellular space. The infection is basically taking place through one arthropod, which I will show you in a short while, and there is a two ways by which this parasite is transmitted inside the human host. One is a saliva type, okay, which we call it as a salivae variant, okay. So when the when this arthropod bites, okay, then this releases the saliva, and this saliva is loaded with this parasite, and this is transmitted to the human host. Another is when they take the blood meal, okay, then they take a lot and then they excrete, they release the fecus, and this fecal material contain, if it is containing this parasite, then this parasite will reach to the human host. So maybe that will cause some irritation, and this irritation may result to the scratching of that skin, that part of the skin, and that will cause the translocation of this parasite, okay. Starcocarian, the technical term is, okay, which is used for the transmission of this parasite through fecal contamination, okay. So African sleeping sickness is basically transmitted through this this arthropod, which we call it as a tsetse fly, okay. Here T is silent, just for your information. It is transmitted through tsetse fly, which is a Glossina species, which is found in sub-Saharan Africa. And there are two morphological indistinguishable subspecies of the parasite, okay, cause distinct disease pattern in human. One is that Trypanosoma brucei gambinensis causes a slowly progressing African trypanosomiasis in Western and Central Africa. Another is Trypanosoma brucei rhodesiense, cause more acute African trypanosomiasis in Eastern and Southern Africa, okay. So the clinical symptom is sleeping sickness is divided into two stages. In early stage, here you can see that the parasite can be found in blood stream and interstitial spaces of several organs. So there are spaces between the organ, so over there also they may present. patient experience myriad of symptoms, variety of symptoms include chronic and intermittent fever, headache, pruritus, okay, and there is a lymphoadenopathy, and infrequently there will be a hepatosplenomegaly in early stage. Okay. The parasite actively evade the central nervous system. This is a complicated situation. Okay. So, the parasite actively evade the central nervous system, making the start of late stage, sleep disturbance and neuropsychiatric disorder. So, basically when they enter in the central nervous system, this may cause the one of inflammation of meninges, which we call it as a meningoencephalitis. Okay. So, this can cause the meningoencephalitis. Okay. And this is along with neuropsychiatric disorder dominate the clinical presentation, giving rise to disease common name sleeping sickness. And this is basically at this stage there may be a loss of consciousness also in very advanced stage. Okay. Trypanosoma brucei gambiense infection is a chronic with estimated average duration of around 3 years, evenly divided between 2 stages. Another is Trypanosoma brucei rhodiense. Disease is usually acute and death occur within a week to months time. You can understand this is much more dangerous, possibly due to parasite being less adapted to the human. Okay. Now, I will discuss about the life cycle of this African Trypanosomiasis. Okay. So, during blood meal on mammalian host, an infected tsetse fly, that is genus Glossina, inject this metacyclic tropomastigote. Here you can see that the insect is injecting this tropomastigote into skin tissue. Okay. The parasite enter the lymphatic system, okay, and pass into the blood stream. Okay. Inside the host, they transform into bloodstream tropomastigote. Here you can see it's a tropomastigote. Okay, are carried to the other side throughout the body and reach other body fluid like lymph, spinal fluid, cerebrospinal fluid, if you know, and continue the replication by binary fission. Okay. So, this injected metacyclic tropomastigote transform into blood stream tropomastigote, which is carried to the various sites as I have explained you. It can be present in lymph, it can present in cerebrospinal fluid. Okay. And over there they will go multiple this binary fission division. Okay. The entire life cycle of this African trypanosoma is represented by extracellular stages. The tsetse fly become infected with bloodstream tropomastigote when taking blood meal on infected mammalian host. Here you can see that this is these tropomastigote, and this is taken up by the tsetse fly. Okay. In flies midgut, here you can see now it is in the life cycle in the fly. Okay. In flies midgut, the parasite transform into procyclic tropomastigote. Here you can see that they are differentiating into the procyclic tropomastigote multiply by binary fission. Okay. Leave the midgut and transform into epimastigote. Here you can see this is transformed to the epimastigote. Okay. The epimastigote reach the fly's salivary gland and continue multiplication by binary fission. Okay. Here you can see that this is keep on multiplying and making the tropomastigote. Okay. The cycle in fly takes approximately 3 weeks. Rarely for Trypanosoma brucei gambicinae may be acquired congenitally if the mother is infected during the pregnancy. Okay. So, this is another new piece of information. Okay. So, Trypanosoma resistance to the mammalian immunity. Okay. When parasite is phagocytosed, the stress induced activation of adenylyl cyclase of the Trypanosoma plasma membrane leads to the parasite release, that is cyclic AMP into myeloid cell, and constitute consecutive activation of protein kinase A. Okay. And inhibition of TNF synthesis, thus preventing the immediate elimination of Trypanosoma by the mammalian host. Okay. In the mammalian host, the whole parasite is covered with a coat of about 10 to the power 7 identical molecule of glycoprotein, which we call it as a VSG. Okay. This is variant surface glycoprotein. This is very important, just remember that thing. Okay. Which is anchored into the cell membrane via glycolipid, okay, and glycosyl phosphatidyl inositol. Okay. Parasites are able to frequently switch their glycoprotein coat. This is very important, just follow it. Okay. Continuously making the antibody response of the host obsolete and in evading clearance. Okay. I will explain you in more detail in subsequent slide. So, here I just want to explain you that they have about 1000 to 2000 copy of this VSG protein, okay, that is variant surface glycoprotein, and at a time only one expresses. Okay. So, our immune system, here I just want to say that our immune system is quite efficient in clearing this parasite. However, our immune system cannot clear 100%. Okay. Still 1% will be remain there, or a very small fraction will be there, and that small fraction they will change this surface this variant surface glycoprotein, and as I told you, they have 1000 to 2000 copies of this gene. So, there will be a translocation of this VSG at expression site, and then that previous protein will be replaced. Okay. So, in that way they keep on changing the surface. In that way they can very efficiently evade the immunity or antibody mediated immune responses. So, this is the strategy by which this Trypanosoma brucei successfully evade the immunity. Okay. So, drugs against Trypanosoma brucei, it is usually recommended that Trypanosoma should first be removed from blood and lymph by use of these are the drug suramine and pentamidine, and subsequently from the extravascular spaces, that is central nervous system or cerebrospinal fluid with these drugs. Here you can see that milasoraprol, okay, and other drug includes eflornithine and nifetromox. Okay. So, these are the drugs against this Trypanosoma. Okay. Here again I am showing a very interesting video, okay, which is explaining the life cycle of this parasite in in human host as well as in tsetse fly. Okay.
The life cycle of Trypanosoma cruzi in the human host. In this video, we see the life cycle of Trypanosoma cruzi in the human host. The trypanosome life cycle begins with the triatomine insect, which is infected with T. cruzi, biting a human host. Following the ingestion of blood, the metacyclic trypomastogotes are released with the feces from the insect near the area of the bite. Note that this form has great motility. Observe that the parasite enters the host through the bite site when the host scratches the site or when the parasite moves to a region covered by mucosa. The parasite reaches the mammalian bloodstream and subsequently invades different cell types that it encounters, such as macrophages, muscle cells, epithelial cells, and neurons. In this example, the attachment of the trypomastogote form of the parasite to the macrophage surface is observed. The process of internalization via phagocytosis begins with the formation of pseudopods. During and after the internalization of the parasite, the parasitophorous vacuole assembles. Host cell lysosomes migrate toward the internalization region and fuse with the parasitophorous vacuole. The lysosomal content is released in the vacuole. However, the parasite is not affected. In the vacuole, the trypomastogote transforms into an amastogote. This transformation is accompanied by the disruption of the parasitophorous vacuole membrane. The amastogotes are released into the cytoplasm of the host cell and divide multiple times. Observe how many amastogotes are accumulating through the division process. They can occupy the entire cytoplasm of the host cell. Following division, the amastogotes transform into trypomastogotes, which show intense and constant movement that culminates with their bursting out of the cell. The trypomastogotes reach the extracellular space and, subsequently, the bloodstream. The parasites will now infect new cells.
Now here you can see that the life cycle in the tsetse fly. Okay.
The life cycle of Trypanosoma cruzi in the invertebrate host. In this video, we see the life cycle of Trypanosoma cruzi in the insect. The life cycle of Trypanosoma cruzi in the invertebrate host begins with the ingestion of trypomastogotes present in the blood of the vertebrate host during a blood meal. In the stomach of the insect, trypomastogotes transform into epimastogotes and spheromastogotes. Note that some of the parasites in transitional forms, epimastogotes and spheromastogotes, are lysed in the stomach. Epimastogotes pass through the stomach to the intestine, divide by binary fission, and attach to the perimicrovillar membranes of the intestinal cells. Note that this adhesion occurs predominantly through the region of the flagellum. Epimastogotes are released and travel to the rectum. At the most posterior region, many of the epimastogotes transform into metacyclic trypomastogotes and adhere to the cuticle lining, the epithelium of the rectum, and the rectal sac of the insect. When the parasites leave the epithelium, the metacyclic trypomastogotes may be eliminated in the urine or feces of the insect. During subsequent insect bites, metacyclic trypomastogotes infect the vertebrate host, starting the cycle over.
So, with this I am completing the parasite infection as well as this whole syllabus of host pathogen interaction, as well as we have studied the deep immunology, we have studied innate immunobiology, adaptive immunobiology, organs, various things. And I hope you have enjoyed this course, and I am looking forward to see that you like this course, and it is quite helpful in learning our defense system. Thank you very much.